we had 5,000 patients in our cohort, and probably per patient, we were spending, $9,000 to $10,000 to quantify all of those different things at every single level. And that's just really unlikely to be used in any type of healthcare context. It's way too expensive. So the other thing that we did is we knew we wanted to take all of this detail, all of this information from multiple omics, but we also knew we had to consolidate it into a test which could be done at a much lower price and at scale. And so we chose DNA methylation as the one level because that's obviously our expertise to do that. And so now with DNA methylation, we obviously are predicting this omic age, which has been trained off of so much information. But we've also been able to have some other unique tools and tricks which can actually use DNA methylation to quantify things previously you could only do through advanced proteomics or metabolomics.
Right. So let me jump in there and just break down some really important parts of what you just said, because this omic data you were describing at the opening of the interview is this rich source of information from all different systems in the body. And what you were just saying is that it's very expensive to measure those things directly, but they are very, very indicative of aging outcomes. So we want that information. Now, what True Diagnostic and your research collaborations have made possible is using the DNA methylation data to make estimates or predictions of those, for example, blood markers or other parts of the omic information. So you're actually able to go the other direction and point at what's happening in the bodily systems that those omic areas represent with some really interesting accuracy. I'll use that term. I know it's early days.
Yeah.
That gives us information about why an individual gets the age result that they have. And that's a really big advancement.
Yeah, no, I think that's exactly it. I think that we're starting to understand the why of biological aging by incorporating more detail about the things that are driving the process. So in our study, that was actually the first step that we did is using methylation to predict other values. So from now, right now we can use methylation to predict things you would get at a typical doctor's visit, things like your HbA1c for blood sugar management, or your fasting insulin, or your fasting glucose, or your creatinine or kidney function markers. And so now we can use DNA methylation to output these things that previously were things you had to go get a blood draw at your physician's office to actually quantify. And so that was really the first step in this analysis. But then we sort of asked the larger piece is if we wanted to predict outcomes, so particularly if we wanted to predict death, which of those was relevant? Which of those predictions could actually help inform us about the biology of aging? And together that sort of created this omic age, which can obviously quantify your aging, but now also give you the why. What's driving that process in you individually that might be different than driving the process in anyone else.
Right. And to that point, this ability to use the methylation data to predict different biomarkers and provide information about the why, you have already done that work. There are new reports that are going to be accessible to True Diagnostic customers come on the 12th of October, which is really exciting. And moving ahead, this is going to become your primary biological age clock. And this information about these biomarkers, certain selected ones, will be in the reports. And you've offered to show us a sample report with some of this information. So let's pull that up now and walk through some specifics of what information we're able to predict about different omic areas of the body with this DNA methylation data.
Yeah, certainly. So this is a very robust report, and I won't take you through everything, but I do want to just really quickly show some images to help maybe understand the omics. So this is sort of our omic diagram, which talks about those different layers and how we would classify it.